All right, so we're going to talk about today muscle physiology. And we're going to start, when we talk about muscle physiology, we're really going to talk about two different types of muscle. We're going to talk about skeletal muscle and smooth muscle. There are three types, skeletal, cardiac, and smooth, but we're going to come back and talk about what's going on with the heart at a later time. So let's just kind of look at this. The skeletal muscle is over here, and it's also called striated muscle sometimes. And the striations are this direction, these bands that you can kind of see in this picture and drawn into this this picture here. And those bands are the striations, but if you can kind of imagine, the whole idea of a muscle contraction is to pull those bands closer together. And even though they just move a little bit, there are so many of them that when compounded, that's what causes your muscle to shorten. That's what causes a contraction. Okay, so we don't see that on smooth muscle. We don't see those. We don't see any kind of banding patterns, and it's because they're not organized in that way. Muscle or smooth muscle tends to. I don't want to say it's disorganized, but it's organized in a way that makes it more capable of surrounding like hollow organs in kind of, it goes around your blood vessels, and it's found in your intestines, things like that. And so, and its activation is more of a, it's turned on for a while, turned off, while skeletal muscle can be activated and then you know, in kind of a twitch fashion. And we'll get into what all that means as we go along. Okay, so again, here's your skeletal muscle, and we see the striations up here. And then if we look over here, we can see why they're striated. Okay, and so what this is, and we'll define it a little bit better later, but it's called a sarcomere, and this is kind of the basic unit of the muscle. So if we kind of zoom in and look at how it kind of slides together, you can kind of see these thick filaments, thin filaments, which we'll talk about what those are made of, and they kind of squeeze together. But that's what makes up those striations. Let's see, it's multinucleated, so that means if you look at a muscle cell, then it has, I guess we can't see it here, we can kind of see it here, it has nuclei just kind of scattered around throughout. Okay, so it's not, most cells have one nucleus and that's it. Muscle cell or striated muscle or skeletal muscles have are multinucleated, which is different from smooth muscle, which only has one nucleus per cell. It contains mitochondria. Now remember, the mitochondria is the powerhouse of the cell. We all like to talk about that, but what exactly does that mean? Really, it means that it uses oxygen, O2, to produce a whole lot of oxygen and glucose to make a whole lot of ATP. Like for every glucose, it makes like 34, 36 ATP. Okay, so that's what it's doing. And the ATP, we're going to see that ATP is the important part, or that's the energy source for a muscle contraction. So the more mitochondria you have, the more strength you're going to have, the more endurance or whatever that you're going to have for a muscle contraction. Okay. Also, the more oxygen and the more glucose you have. So where that matters is that the number of mitochondria that you have is not like programmed. It's based on demand. So if you exercise more, if you have, you know, intense regular aerobic exercise, or like running, like if you want to run a marathon, something like that, then as you train at first, you know, you're not going to have as much of that endurance, and then over time the number of mitochondria that are found inside your muscle cells will increase, so you'll have more blood flow to get oxygen and glucose there, which is why people may start out barely being able to run a mile, and then in 6 months time, you know, they can run, you know, a marathon or whatever. Okay. Skeletal muscle works mainly in a voluntary manner, so that's also different from smooth muscle. Smooth muscle is it's involuntary; it just kind of does it on its own. Skeletal muscle, you can turn it off and turn it on. That doesn't necessarily mean that you always do. Breathing is a subconscious thing. It's not something we think about, but the thing is, if you want to take a breath, you can. So you do have voluntary control over it, even though it's kind of automated based on, you know, and your breathing rate can change based on, you know, how much carbon dioxide or oxygen is in your blood. but the fact that you can control it means that it's skeletal muscle. So there's always a nice little question about the diaphragm, the thing that's pulling down on your lungs. Your lungs don't have any muscle tissue. Your lungs are just bags. But the diaphragm is the muscle that pulls and causes them to expand. Is the diaphragm skeletal muscle? It's not really attached to a skeleton, but yeah, it is. It's considered the same type of muscle. It's striated and it's under voluntary control. All right, so depolarization of the muscle cell causes cycling of the basic contractile units called psychomeres, and we're going to spend like 6 slides talking about how that works. And calcium, we're also going to spend a lot of time talking about how calcium plays a major role in muscle contraction. So little bits and pieces of all of these, this picture and the other pictures are going to show up, and we're going to, when you look back, you're going to know all of that stuff. All right, so some more facts about skeletal muscle, in case it's, you're ever playing Trivial Pursuit or, are taking a test and these questions are on it. The number of skeletal muscle fibers is set before you're born. So we talk about that with neurons, and we know that neurons, you're born with a certain number of brain cells, and then that's all you have, and brain cells don't divide, which is all mostly true, but it's also true with skeletal muscle. So skeletal muscle fibers are set before you're born, and most of these last a lifetime. So if you think, well, when I was a baby, I was a lot smaller and my muscles were little tiny things. Well, muscle growth occurs by hypertrophy, which is the increase in size. So an enlargement of the existing muscle fibers. And then shrinking occurs by atrophy, which is getting smaller, and if it gets to that point, then they could actually atrophy and die. Okay, your body will just say, okay, we're not using these, and that happens a lot of times with people who are paralyzed, have lost function of their muscles, because the body's not going to spend a lot of resources taking care of something that's not being used. All right, testosterone growth hormone can stimulate hypertrophy. Let's see. And that's again that growth. All right. So what makes up a muscle? Proteins. So we talk about meat being high in protein. Well, it's high in proteins because that's kind of what the muscle is made of. It's made of a bunch of, it's filled with a bunch of proteins that are primarily there for contraction, to cause the contraction. So we have thick filaments and thin filaments. In most books, and even us here, we'll talk about thick filaments versus thin filaments. The thick filaments are made of myosin. Myosin is a motor protein, and so it's a motor protein that is shaped as such, and when it is activated, it pulls. Okay, so it kind of moves in this direction, and it will pull. So if you can imagine that if it's attached to something here, that when it pulls, when it has this power stroke, it's going to move forward. Okay, so if you take your hand and you kind of bend, you put your hand on your palm and then you bend your fingers, it'll pull on your other hand or your other arm. Okay, so if you can kind of imagine doing that, and that's how a muscle contraction takes place. It's just thousands of them all at once doing it, even though they're very small. So myosin is that motor protein, and then it will bind to actin. So in this little picture up here, then I could say the actin filament is actually down here. Okay, and so that's what it's pulling along. It's pulling along that actin filament, and if that actin filament is kind of stuck in one place, then the myosin is going to move forward. Or if the myosin is stuck in one place, it's going to pull the actin together, which is actually what happens. Okay, so that'll make more sense in another picture. So the thin filament is made of actin, which is this chain or whatever that I just drew there, so it's actin, and then on that actin it has something called troponin and something called tropomyosin. And that together those make up the thin filament. So let's talk about troponin. Troponin is actually made of 3 other proteins, and you can kind of see that. They kind of tried to show that in this picture here. So there's the troponin, and it's got the I for inhibitory, C for calcium, and T for tropomyosin. And they all kind of do those different things. So troponin T attaches to the tropomyosin. And let's just look at tropomyosin. Tropomyosin is kind of going around the actin. Okay, so it's kind of doing a kind of spiraling around the actin. And what it's doing is it's blocking the ability, or not, blocking the ability for myosin to bind to it. Okay, so the myosin chain, remember we're saying the myosin chain is going to attach to this and then pull it. Okay, but if that tropomyosin is in the way, then it's not able to do it. So it can only bind if the tropomyosin is moved out of the way from the binding site of the actin. Okay, so that's the myosin, and then it's got to bind somewhere on that actin. So so the troponin. Whoa. Sorry, my whole screen just moved. That's not good. Not good at all. Right. Sorry about that. I don't know how it happened, so I'm having a hard time moving it back. Okay, there we go. Okay, sorry, I didn't want to start all over again. Let's see. So let's see. We were talking about troponin, the 3 subunits, and so the troponin T is the part of the troponin that binds to the tropomyosin. You can kind of see that in this picture down here. Troponin I is what is going to cause that inhibition of the binding of the actin and the myosin. By moving that tropomyosin out of the way, and then, or pulling on it. And then troponin C is the calcium binding. So remember I said calcium played a big role in muscle contraction, and so calcium binds to the C subunit of the protein, the protein subunit of the troponin. Okay, so it's got those 3 subunits, and then we'll just keep going over what everything is doing. There are some other structural proteins, titin and dystrophin. I put up here a picture of dystrophin. Dystrophin is kind of named that because of muscular dystrophy. So the dystrophin is the protein that can be, a lot of times it's a genetic mutation, but it's the one that is damaged in muscular dystrophy that they found. So they found that this, and what it does is it kind of anchors through this whole huge protein complex. But the dystrophin is this one right here, and it kind of helps anchor the muscle fibers. There's an actin, helps anchor that muscle fiber to the membrane, so the cell membrane, which is called the sarcolemma, but it's the plasma membrane in a muscle cell. And that's what kind of holds everything together and keeps that cell stable. And so when the dystrophin is destroyed, then that muscle will fall apart and the muscle cell will die. Okay, and that's what happens in muscular dystrophy. All right, other structural. Let's see. So titin is another one that we'll see. We don't have a picture of it here, but we'll see it. And it's just a structural thing. Holds that myosin kind of in line. So they hold the filaments together with dystrophin anchoring it to the cell membrane. All right, so again, this is kind of a zoomed in picture of the sarcomere. So the sarcomere is that basic contractile unit. There are a lot of letter classifications, I guess, of the regions, because you can see here the A band, the I band, the Z disc. I'm not worried about, like, in the M line, I'm not worried about saying, okay, label this. The reason we have those is because if you look at it through a microscope, you can kind of see the banding, and you can see parts of the banding, not just, you know, dark and then light, that you can actually see these little parts of it. So they named them. What we care about is just using them as reference points to kind of explain what's going on. So this second paragraph here, second bullet point, a contraction happens when Z discs are pulled closer together. So I put these arrows up here saying, so if this is the Z disc, that being pulled toward the middle on both sides is what a contraction is. And then you just multiply that times all of the sarcomeres that are in the cell or in that area. Okay, so the question then is how does that happen? And that's what we're gonna learn. Oh, also there's the titin, kind of again structural, holding that myosin together. All right, so the contraction process is initiated by calcium. It is ended by calcium leaving. Okay, so the overlapping thick and thin filaments in each sarcomere move past each other. So again, you can see our myosin. So the little things up here are the myosin light chains, and then the big one down here is the myosin heavy chain. But you can see the myosin is going to walk along those actin filaments. Okay, so they're gonna walk just like just bending back and forth and attaching and then pulling themselves along the actin, and that's going to cause that actin and the Z disc to pull in that direction. So these are stationary. The myosin is stationary, and then the Z discs move in. Okay, and that's what we're after, and that's going to be shortening. So they move past each other. So that sometimes we call that a sliding filament, but it's not really sliding, is it? It's walking. Okay, and another picture of that is down there, where you can see the myosin, and then it moves this direction, and then it goes back, and then it detaches, and then it goes back, and then it moves, and then it detaches, it goes back. Okay, so that's called cross-bridge cycling, which we're going to have a slide on that too. So the ability of a muscle fiber to generate force and movement depends on ATP-powered myosin motor proteins. So these motor proteins aren't walking just out of the goodness and kindness of their heart. They do it because they are powered by ATP. Okay, so ATP is the source of energy for those myosin motor proteins to walk, to move. And of course, that's why walking gets us tired, because it burns ATP, and then we need glucose and oxygen to continue to make more ATP and regenerate that ATP. Okay, so that's why we get tired when we run or walk even. All right, so we're gonna talk about a depolarization event. So we have motor proteins, or I'm sorry, not motor proteins, but motor neurons that are going to send a signal saying, hey, it's time to contract. So an action potential moves down a neuron. So we can draw one here if we want to, because this picture doesn't have one. This is from your book. Okay. And it's going to release acetylcholine onto nicotinic acetylcholine receptors. Okay? And that's going to generate an action potential. It's going to let sodium in, and then it's going to depolarize, and then potassium is going to move out, and it's just going to be all the way along. Depolarization. Well, we want it to be able to get down deep into the muscle, so that action potential is sensed all the way through. Okay, and so we have something called T-tubules, or transverse tubules, which allows it to, instead of just like running across the membrane like that, and the middle being kind of left out of the whole thing, it moves in. Okay, so that's what the T-tubules are. So they're invaginations of the sarcolemma, invaginations of the cell membrane, the muscle cell membrane, and that action potential then can move along those. All right. So the muscle action potential travels along the cell membrane and the internal membranes called T-tubules, transverse tubules. This activates calcium release from ryanodine receptors. We're going to see some of those. So our calcium is stored in these guys, which this is, it's not the endoplasmic reticulum, it's a modified endoplasmic reticulum found in muscle. It's the sarcoplasmic reticulum. Okay? Reticulum. And that stores calcium. Okay? So lots of calcium inside this. And so when the action potential moves down into this area, it starts letting calcium out of the endoplasmic reticulum. Okay? And once you have calcium out of there, as long as you've got ATP, you're going to have a muscle contraction. Right? So calcium stored in the sarcoplasmic reticulum is released. Remember that subunit on the troponin called the troponin C? The calcium binds to troponin in that area, which causes it to move tropomyosin out of the way, which triggers that cross-bridge, something called cross-bridge cycling, which we're going to talk about in a couple of slides. Okay, so we're getting somewhere now. We've got a signal of a motor neuron coming in and causing depolarization. The depolarization moves down the T-tubule, and then you have something called a DHP, dihydropyridine or something like that, and ryanodine receptors that are going to allow calcium to move out of the the sarcoplasmic reticulum, and then you have a muscle contraction. Okay, so let's kind of look at that. So here is a blow up of the DHP and the ryanodine receptor. So this is kind of the same thing, just a little bit different perspective. So the sarcoplasmic reticulum in muscle is homologous to the endoplasmic reticulum found in most cells. Calcium is stored and released following membrane excitation. I also say that calcium is stored in the endoplasmic reticulum of most cells too, by the way. So there's the calcium stored in the ER, and then we've got the microtubules. They're not showing them here. Microtubules and actin kind of spread all through there too. You have motor neuron, depolarization, moves down into the T-tubules. Okay, so we saw that on the other slide. But now we can kind of see this DHP and ryanodine receptor. The DHP receptor, let's see, it's talking about it here, is sensitive to that depolarization, that change in membrane potential. All right, so it is bound to, connected to something called a ryanodine receptor, which is associated with the calcium channel. Okay. So the DHP receptor activates the ryanodine receptor. So the DHP receptor is the voltage meter. It activates the ryanodine receptor, and the ryanodine receptor causes that calcium channel to open, and then calcium is released. Okay? And then calcium binds to troponin, which moves tropomyosin out of the way, so the myosin heads combine. So we can see that picture here. Okay? We see the troponin here with its three little subunits. The calcium will bind to that troponin, the tropomyosin, which is kind of swiggling along here and blocking the myosin binding sites, so it's kind of in the way. But the calcium, when the calcium binds to that troponin, it moves that out of the way. So then it exposes the myosin binding sites, so then the myosin can attach, and as long as you've got ATP present, it will start to plus PI, hydrolysis of ATP will cause this cross-bridge cycling to happen. Okay. All right, so I like this picture because it kind of shows, it makes an attempt to show the myosin binding site. So the binding of calcium here to the troponin. So right now, this little rope-looking thing, this little blue rope, that's the tropomyosin, and it's blocking the myosin binding sites, which are kind of these dark purple areas. Okay, so it's blocking those, and so the myosin is unable to attach to them. Okay, so no attachment can't take place because they're blocked. All right, so once we have calcium present down here, then it moves that tropomyosin out of the way. So now you can see these are fully exposed. These myosin, or the myosin binding sites are fully exposed because the tropomyosin has been moved out of the way, because the troponin pulled it out of the way after calcium was bound. And now we have myosin is able to start binding and start walking along that actin filament. Okay? So we have, again, calcium is released from those ryanodine. Ryanodine receptor opens that calcium channel. Calcium is released, binds troponin, moves tropomyosin out of the way, exposes those myosin binding sites, and now we have cross-bridge cycling. So let's talk about cross-bridge cycling. After calcium, after tropomyosin is moved out of the way, as long as you have both ATP and calcium present, you're going to have this cycle that takes place. Okay. The cycle starts, we like to start the cycle here, and that is with the myosin head bound to the actin. I have this in purple. So when the cycle stops, the myosin stays bound to the actin. Now, usually in a cell, there's ATP present. Just ATP is just always present. Most of the time it's present. Unless you've used it all up. So ATP will dislodge that. So the binding of ATP to myosin, okay, so we can see it bound to myosin in this second picture, will cause the detachment of the myosin head from the actin filament. Okay? And when that happens, now it's released. So the next step, so we have ATP bound to, physically attached to the myosin head, and then we have hydrolysis of the ATP. Hydrolysis of the ATP means that it is broken into ADP, adenosine diphosphate instead of triphosphate, and that inorganic phosphate group is separated. So it's cleaved, it's hydrolyzed, and separated. And when that happens, that's a process that releases energy, and because sticking that last phosphate group on requires energy, so when you pop it off, it releases energy, and what it does is it transfers that energy to the myosin head and energizes it. Okay, so it's like pulls it like it's like pulling a rubber band or something, and it's putting it over there, and now it's energized and it's just ready to go. So it's almost like cocking a gun or something. It's out back there and ready to go. And so then all it needs is something to allow it to go. And really, since you've got calcium and you've got ATP, you still have calcium present, whatever, it's going to automatically then attach right after it is energized. It's going to attach to the actin and immediately pull. Okay, so it changes, and so this is cocked and ready to go, and then it will pull on it and go to this position. Okay, so if you compare these positions, you can see that this one is the energized position, and that one is after it's kind of moved and pulled the actin filament, and then it's stuck there. So I guess the ADP, the inorganic phosphate falls off when it binds, and the ADP falls off after it pulls, whatever. But now it's sitting there with no ATP, no ADP, nothing on it, still stuck, bound to the actin. So if we come back over here to our list here, so when that cycle stops, myosin stays bound to the actin, and it won't release until a new ATP displaces it. And I have that little question of how is this related to rigor or rigor mortis? So rigor mortis is what happens when someone dies and their body becomes stiff. Okay? It becomes stiff because the person has stopped making ATP, and that ATP gets all used up, and once it's gone, which happens moments after death, then what you find is your muscles have all the myosin heads stuck to the actin. Okay? And that's rigor mortis, and it's difficult to move a muscle when it's like that. Now, after a little bit more time passes, then the muscles there's some breakdown of the proteins, and you're able to move the muscles again, but that initial time is that during rigor mortis is when the ATP is there, but the muscles are still there and bound. Okay. All right. So if we go through this again, so there it is. The myosin head is stuck to that actin filament. Let's say calcium is still present, so the tropomyosin is still out of the way. Another ATP is going to come in, dislodge that myosin from the actin, and really, as long as there's ATP present, it'll sit in this conformation. And now we have ATP on it, but it's not energized, so hydrolysis of the ATP is the next step. Hydrolysis to ADP and inorganic phosphorus, PI. And that's energized, so that's the energizing the myosin head, and then binding to it and the power stroke. Okay, the power stroke is when it pulls it, right? And then the ATP is gone, ADP is gone, everything's gone again, it's stuck to it, another ATP, hydrolysis, energizing, and then the power stroke in these two steps, the attaching and then pulling. Okay. And it just keeps doing that, just keeps doing it as long as you have ATP. Now if you get to the point where you've run out of ATP and you've just exhausted yourself, then your muscles are going to stop working. They're like not going to cooperate, and that's rigor. Okay, so if we put it all together, I like this slide because it has everything. If we, well, okay, it doesn't have cross-bridge cycling, but if we put it all together, we have our motor neuron releases acetylcholine, okay, onto nicotinic acetylcholine channels or receptors, nicotinic acetylcholine receptors on the motor end plate, which causes sodium to rush in and depolarizes the cell, okay, and causes that action potential. The action potential moves down the T-tubule. It is sensed, that voltage change is sensed by the DHP protein in the membrane, which will activate the ryanodine receptor, which allows calcium to move out of the sarcoplasmic reticulum. The calcium binds to the troponin, which moves tropomyosin out of the way to expose the binding sites on the actin, and then the myosin will bind to the actin, and as long as ATP is present and while the calcium is present and that binding site is exposed, you'll have that cross-bridge cycling, which is the contraction. Okay, and we stop a muscle contraction by pumping sarcoplasmic endoplasmic reticulum calcium transporter. So it moves calcium back in. I think that's what that stands for. I don't know. But anyway, it's the calcium transporter. Calcium transporter will pull the calcium back in to the sarcoplasmic reticulum, and then the calcium's gone. Okay? And that's it. And your muscle contraction is over. And we'll talk about that. As long as you're depolarizing it, you're going to keep that calcium in there, and it's going to keep that muscle. Repeated tetanic stimulation is going to keep it contracted. Okay, so we'll get back to that. All right, so let's talk about muscle tension. We just talked about the action potential here, and we know that the action potential happens first. That's what's moving down through the T-tubules, the DHP receptor or DHP protein, activating that ryanodine receptor, and then opening the SR to let calcium out. So if the action potential happens first and very quickly, then calcium release is going to happen next, right? Because the calcium is still stored away in that sarcoplasmic reticulum, Ca2+. So that happens next, and the calcium levels start to build up. Well, then once the calcium is there, binds to troponin, moves tropomyosin out of the way, and lets myosin bind that actin and cause that cross-bridge cycling, then that's when you have actual tension. Okay? So you've got a little time between the release of the acetylcholine and depolarization to the calcium levels building up to the tension that you actually feel. Now it's very rapid, as we know. We can activate a muscle very quickly. If you've ever, seen something like a cat jump or something, that can happen very quickly. All right, so a twitch is a single action potential resulting in a fixed amount of calcium release. So a twitch happens when you release a vesicle. So a vesicle containing acetylcholine is released onto onto an end plate, muscle end plate, or at the neuromuscular junction, and causes an action potential to very quickly move down into the muscle. Okay, so just activation, very rapid activation of that motor neuron will cause a twitch. Okay, and then very, very quickly the calcium transporters pull that calcium back out, and the calcium is gone, and you don't have a muscle contraction anymore. That's a twitch. Now, an actual muscle contraction, sustained muscle contraction, happens from repeated twitches. Essentially, let's see, we'll kind of draw it like that. The motor neuron will continue to, so the brain is saying, okay, we're going to keep this activated, so it continues repeatedly to depolarize. So activity of that motor neuron repeatedly, 100 times per second, is a tetanus or tetanic stimulation, and that's what's going to keep your muscle contracted. All right? Okay. So a tetanus or a fused tetanus. Fused tetanus is kind of more of a smooth type of contraction. Unfused just kind of means that you're kind of shaking, and there's some repolarization occurring in between. I think, but we're not too concerned about that. Fused tetanus is it just means that you've got a continuous muscle contraction. All right, an isometric contraction occurs when the muscle contracts but doesn't shorten. So that means that you're still, like if you're holding something in your arm and you're maintaining the same amount of force, but you're maintaining the amount of force to kind of hold it in that one area. So if you're just holding your hand up, or if you're holding a book in your hand, but you're not shortening the muscle. That's isometric. Isotonic is when you're changing the muscle length, but you're maintaining the same amount of force. Okay, that's an isotonic contraction. These are just terms. The greatest tension. Okay, so this one, I like this point, the tension versus length, because we can relate that right back to our actin and our myosin. So the greatest tension occurs in a muscle length that allows for the maximum overlap of the actin and myosin. So remember, you've got your myosin chains, and then you have your actin, lots of actin. I'm sorry, you've got your actin filament, and then you've got your myosin chains that are walking along that. Okay, so if you've gone too far where you have, actin or myosin that's clear up here, kind of falling off the edge on this side, or just jammed up when the muscle is too short, you're not going to get a very good contraction. Or if the muscle is extended out too far and you've just got myosin kind of dangling there, just not walking on anything because the actin ends there, then that's also not going to be very helpful. So we get to this middle point right here where you've got maximum binding of myosin to the actin filament. Okay, so that's where you have the most tension that can be generated, is at that point where you've got most of the myosin pulling along the actin. You've got good contact, and it's not kind of pushed in too far, which is what happens. Here. So this is when it's a very short muscle and it's kind of jammed up, and then kind of when it's longer, you have good contact. That's where you have this. And then if you extend it out too far, you're really not going to get any kind of tension, you're going to have maximum tension because you've stretched the muscle out just as far as it can. And that's really what this green line is showing. It's saying, okay, we're stretching the muscle out, which, if you stretch a rubber band, it's going to resist that, and so that's the tension, which really, I don't know. I kind of don't think that counts. So I more focus on this active tension than this, just the fact that we're overstretching our muscle. But the graph shows this, what's called passive tension. That's the stretching. And then it kind of adds them together for total. But either way, once you've stretched your muscle out so far, your tension is off the charts because you can't do any more. It's not going to go any farther. All right, so and I have this question here. What muscle length do you feel you have the greatest strength? And it's probably going to be somewhere in the middle, not when it's all fully contracted and not when it's fully stretched out. Okay, so I like this picture because it shows what we're talking about. Okay, so this one you can kind of see things are bunched up over here in A. You can kind of see things are bunched up there. And there's really not much tension you can even form because there's no more pulling that can be had. And then as it extends out, you kind of get to this point where you have where you're not pushing, it's not bunched up, but you have perfect, great contact between the actin and the myosin. So all those myosin, nothing's dangling, nothing's pushing up against. It's just this nice area where you have good contact of the actin with the myosin. And then as it stretches out more, you can see that some of your myosin is just kind of dead out here. It's not doing anything because there's no actin to bind to because you've stretched your muscle so far. Okay, and then you've got to this point where you got nothing. Okay, you've stretched it so far that you can't form a contraction, which really doesn't happen because there's always some contact. Okay, so that's length-tension relationships, and hopefully we understand how skeletal muscle is working. So we're going to move on to smooth muscle, and there are differences. So I want you to know a lot of the details we just talked about with the DHP, the ryanodine, calcium, ATP, T-tubules, the cross-bridge cycling, all of that stuff. I want you to know all of that about skeletal muscle. We're just going to mention some differences with smooth muscle that are kind of important. So essentially, smooth muscle is spindle shaped, so kind of shaped like maybe that, and with a diameter, it's smaller. It's smaller than skeletal muscle. It tends to be smaller than skeletal muscle fiber. So I mean a skeletal muscle fiber, which is another term for a skeletal muscle cell. But these fibers are one cell can go all the way down from your spinal cord all the way down to your big toe. Okay, that can be one cell, one fiber. That's one of the reasons they can be multinucleated. We don't need that with smooth muscle. So smooth muscle have a single nucleus. So they're smaller than skeletal muscle. They have a single nucleus, and whereas skeletal muscle, you're born with all you're ever going to have, smooth muscle, they can divide throughout the life of the person. Okay, so they can reproduce. You can have division of smooth muscle. You can make new smooth muscles. And so really when you have you've walled off something like an abscess or something like that, or if you've ever heard of atherosclerosis, okay, which is where your blood cells your blood vessels kind of get clogged up with stuff, kind of get clogged up with some fat and junk in there. Well, what can happen is that smooth muscles will actually grow around that and kind of help to wall that off. Okay, so they can kind of form these barriers inside the body, kind of a role that we don't really think of. They're not necessarily contracting, but they are a cell that can divide and kind of move around. So they kind of play that role too. So it's handy that they can divide throughout the life of an individual, because without it, I mean, how would you make atherosclerosis? No, I'm kidding. It's usually a good thing to kind of keep that walled off. All right, so smooth muscle, they have the myosin-containing filaments and the thin actin-containing filaments, so they still have that. That's how they contract, but they're just not organized in the same way. And whereas, remember, calcium binds troponin to move tropomyosin out of the way, that's how skeletal muscle works. In smooth muscle, it doesn't work that way. It works with another protein, another couple of proteins that kind of activate that cycling. So the thin filaments are anchored to either the plasma membrane or to cytoplasmic structures known as dense bodies. Okay. All right, so that's how they're kind of put together, and it allows them to have a different kind of movement, or a different kind of contraction. So it works well for those hollow organs that we think about, like with the intestines and the blood vessels. Okay. All right. So thick and smooth muscle, thick and thin filaments are not organized into those nice little nicely organized myofibrils. There are no sarcomeres, so you don't have the banding that you saw with the skeletal muscle. You don't have that. It's not organized in that way. They surround hollow structures and organs that undergo changes in volume with accompanying changes in the lengths of the smooth muscle fibers in their walls. So the intestines, the stomach has smooth muscle around it. Your esophagus, when you swallow, smooth muscle. Your bladder, detrusor muscle, smooth muscle. So they kind of, that's what we mean by these hollow organs, and of course blood vessels. All right, so let's talk about how it's different in the way that it initiates a contraction. Okay, because ultimately we're going to have cross-bridge cycling, but how it gets to that point is different. So smooth muscle doesn't have the troponin, so there's no troponin in smooth muscle. Instead, it has something called calmodulin. Okay, modulin. Okay, so calmodulin is a protein that has 4 binding sites to it for calcium. Okay. So calcium can bind to it, and when 4 calcium have bound to calmodulin, we start to call it calcium calmodulin because it's been activated, and then active calcium calmodulin will activate those myosin light, or it activates a protein called myosin light chain kinase. Okay, so the light chains are these kind of exposed up here in the head of the myosin, and a kinase, that term kinase, is something that puts a phosphate group on something. Okay, so it phosphorylates. So it phosphorylates something, and phosphorylation is a way that the body has figured out. It happens all over the place. Proteins are activated, lots and lots of many proteins are activated by phosphorylation, and this is just one of them. So this enzyme, this myosin light chain kinase, is a protein that phosphorylates other things. In this case, the myosin light chain kinase is phosphorylating the myosin head. Okay. And when it does that, that's not what it looks like. Okay. So we can say that it maybe does it down here. When it does that, it makes that appropriate or able to bind to the actin. Okay. All right. Which phosphorylates myosin, allowing cross-bridge cycling. So that's when, so as long as it's phosphorylated, it's able to do that. So only the phosphorylated form of smooth muscle myosin can bind to actin and undergo that cross-bridge cycling. And to relax, we need to get rid of the this phosphate group. So we dephosphorylate, and that's done with something called a phosphatase. So there's a myosin light chain phosphatase that will take that phosphate group and remove it. Okay, and that's it. That's the difference. So whereas in skeletal muscle, we had this nice way of having troponin, calcium-bind troponin, move tropomyosin out of the way to expose the binding sites. In smooth muscle, we just have to get the, it doesn't have the troponin, so we just have to make the myosin able to bind to the binding sites by phosphorylating it. Okay. All right, so that's the difference. So calcium, again, comes from the sarcoplasmic reticulum. Extracellular calcium enters the cell through the plasma membrane and calcium channels, and then to relax, that calcium has to be removed. Okay. So kind of the same thing. So here we can see how it happens. So depolarization could open voltage-gated calcium channels. Hormones or neurotransmitters open ligand-gated calcium channels, and then intracellular hormones or transmitters, neurotransmitters can activate this inositol triphosphate or IP3, which also causes calcium release from the SR. These all can cause calcium release from the SR. Calcium mediated calcium release. Either way, we're getting calcium release. Okay, so I'm not going to go through and have you list off all of these different ways. We know that activation is happening by hormones, depolarization, whatever means, and we'll kind of see how those two differ. All right, so smooth muscle activation. Hormones released into the blood have their effect wherever a receptor is made. So here we can see hormones or neurotransmitters just binding the receptor, and then we see that inositol triphosphate, the IP3, causing more calcium to be released from the sarcoplasmic reticulum. Neurotransmitters are released locally from varicosities. We're going to have a picture of that on the next page, but that'll like penetrate into a muscle and activate a whole lot of different smooth muscles in an area. Okay, so that's kind of how we get some of that peristaltic activity, or constriction all along a blood vessel. So local signals like pH and carbon dioxide have a local effect, including voltage changes and intracellular signaling. We have inside the blood cells, or inside your body, we have metabolism going on of every cell, and your blood is trying to bring it oxygen, and is trying to get rid of the carbon dioxide. And so if carbon dioxide levels start to build up, you can actually have relaxation of the smooth muscle in those blood vessels to kind of let them dilate and bring in more nutrients, more glucose, more ATP, or more glucose, more oxygen. Okay, to make glucose to make ATP, and oxygen to also make ATP. All right, to give it more energy, and to bring in more oxygen, and to remove the carbon dioxide. So those are kind of local signaling that can happen. But anyway, all of these lead to an increase in intracellular calcium and muscle activation. All right. So here's a picture of the same point, but you can see these varicosities. So you have an autonomic nerve fiber. So this could be, sympathetic, parasympathetic, either one, but it's going to release the neurotransmitter onto the muscle cell, and you're going to have contraction. Okay, so neurotransmitters released from varicosities on one end, released from varicosities on one axon may activate many smooth muscle cells. Same neurotransmitter, so that's good, because that kind of gets a whole area. Same neurotransmitter may have opposite effects in different areas depending on individual receptors. Think of just the general way that the autonomic nervous system works. Depends on the receptor and what effect it does. So norepinephrine may cause contraction of a vascular muscle, so for vasoconstriction, but may cause relaxation of bronchial muscle, or it may cause contraction of vascular muscle in one area and relaxation in another area. Local factors such as acidity, oxygen, carbon dioxide, osmolarity, ion content, those can all change smooth muscle tension. I want to point out that nitric oxide is an important vasodilator. So nitrates, things like that, can cause relaxation of that vascular smooth muscle, and can help with blood flow. All right, mechanical signals such as stretching may also activate smooth muscle. So where an example of that would be like in the bladder. Okay, and that's it. Have a good day.